Ladies and gentlemen, big major bone defects are still an unsolved problem in orthopedic surgery. We have actually different options like anaprosthesis, but also the disadvantages like loosening abrasion. And we have other options like biologic reconstruction with allografts and artificial bone substitutes. The problem with allografts is, like you can see as example in this case, we can resect and we can put the allograft in, but a couple of years later you see there was no ingrowth of the allograft, and the artificial surface, or the articular surface is collapsed. So advantage of allografts are the availability and bridge covering big defects, but disadvantage is in terms of sterilization and procurement protocols. You have significant alteration of biological and biomechanical properties due to insufficient vascularization, and so we get this kind of fatigue fractures. So it would be a goal to do a kind of biofunctionalization of this kind of allografts, and the goal or the objective of this study was the coating of sterilized bone grafts with some peptides called RGD-peptides to improve cell proliferation. So RGD-peptides are three peptides, and they are present in extracellular matrix like fibronectin and collagen, and they are interacting with the cells via transmembrane protein receptors called integrins. And they work like this. You have the matrix, you have the RGD peptides in the matrix, and via this kind of integrins you get different kind of, or there's transduction or signal transduction of apoptosis, gene expression, proliferation. So in our study we used cyclic RGD peptides, cyclic because it's prevent for proteolytic degradation. They're special anchors, so they we have optimal binding, and this peptides we're using are specific for certain integrins who are present in endothelial cells and osteoblasts. So what have we done? We put out cancellous bone specimen out of femoral heads after hip arthroplasty, several washing steps, thermodisinfection, and 82 degrees, and incubation with this kind of RGD peptides. Afterwards, application of human osteoblast-like cells and endothelial cells for 30 minutes. Afterwards, we seeded cells for 7 days of cultivation, and we have done a quantification of cell amount 24 hours after and 8 days after. Plus, we've done immunohistological staining to evaluate cells after 7 days. As results, we can see here control of titanium in contrast to titanium-coated RGD peptide surface, and you can also already see a big difference in adhesion of osteoblasts. So the difference in adhesion you can't see in bone, because in bone we have still some collagen left, so there's no additional effect of the RGD peptides. In contrast to proliferation, after 8 days, you see to the control, it's up to 40% better proliferation of osteoblast-like cells. So it's the same for RGD peptides, for the HUVEC cells, for the endothelial cells. You have after 8 days up to 30% more proliferation. So conclusion, RGD peptides are inducing more cellular growth and proliferation of osteoblastic cells and endothelial cells. Perspective, we have to get an animal model. It's going on to confirm the preliminary results. Thank you.